Welcome back to Studious, where we explore the incredible biological mechanisms that keep the human body running. Today, we are diving deep into a remarkable tissue that works entirely behind the scenes, smooth muscle. This presentation, titled The Architecture of Involuntary Motion, will serve as a visual guide to smooth muscle histology and physiology. Have you ever wondered how your eyes adjust to light without a single conscious thought? Or how your digestive system continuously processes food long after a meal? This seamless, involuntary motion is the hallmark of smooth muscle tissue. It acts as the silent architect of our internal organs, working around the clock to sustain vital life functions. Unlike the skeletal muscles you flex voluntarily, this unique tissue operates entirely on autopilot. Governed by the autonomic nervous system, it prioritizes efficiency and endurance over rapid voluntary force. Throughout this video, we will break down the microscopic structure of these fascinating cells, explore how they physically contract, and understand how they assemble to power the complex organ systems we rely on every single day.
Before we examine smooth muscle in isolation, we need to understand where it fits within the broader structural framework of the human body. Muscle is actually one of the 4 foundational tissues that make up our anatomy. The other 3 are epithelial tissue, which lines our surfaces, connective tissue, which provides support and structure, and nervous tissue, which handles electrical communication. Within the muscle category itself, we have 3 distinct variations: skeletal, cardiac, and smooth muscle. Despite their differences in function and location, all muscle tissue shares a common fundamental building block: the myocyte. The myocyte is a highly specialized, elongated cell that comes packed with the contractile proteins actin and myosin. These proteins are arranged in distinct bundles that allow the cell to shorten and generate force. Whether it's a bicep curling a weight, a heart pumping blood, or a stomach digesting food, these underlying cellular components remain the universal engine of biological movement.
To easily identify and categorize muscle tissue under a microscope, histologists use a diagnostic matrix that evaluates appearance, control mechanisms, and anatomical location. Let's compare them. Skeletal muscle is attached to bones and tendons. It features a striated appearance, meaning it has visible stripes, maintains a consistent diameter, and has peripheral nuclei. Crucially, it is voluntary, controlled by the somatic nervous system. Cardiac muscle, found exclusively in the heart, is also striated, but operates involuntarily, continuously pumping blood without our conscious input. Finally, we have smooth muscle. Unlike the others, it is distinctly non-striated, giving it a visually smooth appearance under a microscope. It is entirely involuntary, meaning its activity is not monitored or initiated consciously. Anatomically, it is the primary tissue that lines the walls of our hollow organs and internal tubes, such as the intestines and blood vessels. Understanding these core differences is essential for identifying normal tissue architecture in any histological sample.
Because smooth muscle forms the walls of hollow structures, it drives involuntary motion across an incredibly diverse array of organ systems. Let's trace its path through the body. In the eye, the ciliary muscle controls lens accommodation, adjusting our focus for different viewing distances, and regulates the flow of aqueous humor into the canal of Schlemm to maintain ocular pressure. Moving down to the respiratory tract, smooth muscle wraps around the bronchi and bronchioles to constantly regulate the amount of airflow entering the lungs. In the gastrointestinal tract, it supports GI motility, using both tonic and forceful contractions to push matter through the digestive system. Throughout our circulatory system, smooth muscle lines the walls of our blood vessels, regulating their caliber through vasodilation and vasoconstriction, which directly controls our blood pressure. Finally, in the reproductive system, the smooth muscle of the uterine myometrium induces powerful contractions during menstruation or labor. It is truly a multi-system workhorse.
When we look at individual smooth muscle cells, their physical shape is perfectly optimized for their function. They exhibit a fusiform architecture, meaning they are elongated cells with distinctly tapered ends. They are quite small, ranging in dimensions from just 0.2 to 10 micrometers in diameter and 50 to 200 micrometers in length. Interestingly, their exact shape and the appearance of their nuclei will actually vary depending on their current state of contraction. This unique fusiform shape isn't just for show, it enables incredibly dense cellular packing. The cells are arranged in a staggered alignment, where the thick central portion of one cell nestles against the thin, tapered ends of its neighbors. Furthermore, these cells are linked directly to one another via specialized gap junctions. These gap junctions allow for rapid chemical and electrical communication. Combined with the tight, staggered packing, this network achieves highly efficient cell-to-cell force transfer, allowing the entire tissue bed to act as a unified, coordinated sheet.
When examining tissue in the pathology lab, the standard preparation is the H&E stain, hematoxylin and eosin. Identifying smooth muscle relies on recognizing two distinct colorations on these slides. First, we look for the basophilic nuclei. Hematoxylin binds to acidic structures, like DNA, staining the nuclei a dark bluish purple. In smooth muscle, these nuclei are characteristically elongated and centrally located within the cell. Second, we examine the surrounding cytoplasm, also known as the sarcoplasm. The sarcoplasm is eosinophilic, meaning it takes up the eosin dye and stains an even, bright pink. This smooth, uniform pink coloration is specifically due to the incredibly dense concentration of the contractile proteins actin and myosin packed inside the cell. Because there are no rigid sarcomere grids breaking up the protein layout, the stain appears beautifully consistent. For a histologist, spotting this combination of staggered, cigar-shaped purple nuclei floating in an even sea of pink cytoplasm is the classic signature of smooth muscle tissue.
Smooth muscle cells do not operate in a vacuum. They require a robust support system to function. The cellular envelopes surrounding them provide critical structural support and chemical regulation. Right at the cell surface, we have the sarcolemma. The term comes from the Greek sarko, meaning flesh, and lemma, meaning husk or sheath. The sarcolemma encompasses both the plasma membrane of the cell and its external lamina. Moving just outside the cell, we find the endomysium. This is a delicate network of reticular connective tissue that surrounds each individual myocyte. The endomysium plays a vital role by providing the exact chemical environment needed to concentrate generated forces and facilitate smooth contraction. Embedded within this extracellular matrix are fibroblasts. Fibroblasts are the maintenance workers of the tissue. They constantly secrete collagen, glycoproteins, and glycosaminoglycans to maintain the structural integrity of the extracellular matrix, ensuring the muscle fibers have a healthy, flexible scaffold to pull against.
To understand how smooth muscle actually moves, we have to look at extreme microscopic magnification. The core mechanism of all muscle contraction occurs when thick myosin filaments physically slide along thin actin filaments. But how does that internal sliding pull the outside of the cell? Enter the dense bodies. Dense bodies act as molecular anchors. These cytoplasmic densities are stationed throughout the cell and firmly attached to the inner wall of the sarcolemma. The internal actin filaments physically tether themselves to these dense bodies. When the myosin slides against the actin, generating mechanical force deep inside the cell, that force is transmitted down the actin filaments directly to the dense bodies on the cell membrane. The result is that the internal tension physically pulls inward on the cell surface. These dense bodies are essentially the hitches that allow the engine of the cell to actually move the chassis, translating microscopic chemical energy into macro-level physical contraction.
The defining characteristic that gives smooth muscle its name is the complete absence of sarcomeres. To understand why this matters, look at skeletal muscle. Skeletal tissue relies on rigid, linear grids called sarcomeres.
These grids create the visible striations we see under a microscope, and they restrict the muscle to pull strictly in one linear direction. Smooth muscle, however, lacks these sarcomeres entirely. Instead of a linear grid, the actin and myosin filaments anchor to a web-like network of dense bodies scattered throughout the cell. When contraction is initiated, this dispersed anchoring allows the entire cell to essentially wring itself out, compressing inward from all directions simultaneously. It is this complete absence of linear protein grids that creates the structurally smooth appearance under a microscope. This omnidirectional contraction is a masterclass in biological engineering, allowing the cells to squeeze and constrict hollow tubes in a way that linear skeletal muscle simply could not achieve.
When smooth muscle wraps around hollow organs, like the intestines, it organizes into a highly structured dual layer known as the Tunica muscularis. This structure is an architectural marvel designed for moving material. The inner portion is called the circular muscle layer. Here, the smooth muscle fibers wrap like rings around the circumference of the tube. In specific locations of the gastrointestinal tract, this circular layer thickens significantly to form sphincters, acting as physical valves. Wrapping around the outside of this is the outer longitudinal muscle layer. In this layer, the muscle fibers run lengthwise, parallel to the organ tube, and this outer layer is usually thicker than the circular one inside. Sandwiched delicately between these two contrasting sheets of muscle is a very thin dividing layer of connective tissue. This crucial middle layer serves as a highway, housing the blood vessels, lymphatic vessels, and nerves that supply oxygen, nutrients, and electrical commands to the hard-working muscle tissue on either side.
The alternating arrangement of the tunica muscularis is what makes peristalsis possible. Peristalsis is the wave-like movement that pushes food and fluid through our bodies, and it requires perfectly synchronized bidirectional contraction. It relies on two opposing actions working in harmony. Action A involves the inner circular layer. When these circular muscle fibers contract, they act like a drawstring, squeezing the tube and reducing the width of the organ's lumen. This prevents matter from moving backward. Action B involves the outer longitudinal layer. When these lengthwise fibers contract, they pull the walls of the tube end to end. This action shortens the specific organ segment and effectively pulls the walls outward, widening the lumen ahead of the food bolus. By alternating these two actions, pinching behind the matter while shortening and widening the path ahead of it, the smooth muscle can smoothly and continuously propel contents down the digestive tract without us ever having to think about it.
For the complex dance of peristalsis to work, the smooth muscle needs precise, coordinated electrical wiring. This is provided by the Myenteric plexus, also widely known as Auerbachs plexus. This dense network of nerves acts as the localized brain of the gut. It is strategically embedded right inside that thin layer of connective tissue that separates the inner circular and outer longitudinal layers of the tunica muscularis. From this central vantage point, the plexus sends its axons spreading out caudally, primarily to innervate the inner circular muscle layer. Because it lies directly between the two working muscle sheets, it can perfectly time their alternating contractions. The myenteric plexus serves as the main control center for the motility of the intestinal wall, receiving autonomic inputs and translating them into the rhythmic, synchronized contractions required for healthy digestion. Without this intricate neuronal wiring seamlessly communicating with the smooth muscle, our digestive system would simply grind to a halt.
In biological histology, there is a golden rule: form always follows function. The engineering of peristalsis perfectly demonstrates this concept at three distinct levels. First, we have staggered cellular packing. When individual myocytes align their tapered ends against their neighbors' thick centers, enveloped in a force-concentrating endomysium, it creates a seamless, uninterrupted transfer of power from one cell to the next. Second, we have omnidirectional force. Because the cells lack rigid sarcomeres, they aren't restricted to linear twitches. Instead, they can sustain prolonged inward squeezing from all directions. Finally, this microscopic architecture perfectly serves the macroarchitecture, the dual-layered cylinders of alternating circular and longitudinal muscles. The cellular design makes the tissue efficient, while the organ-level design channels that efficiency into physical waves. Together, this multi-tiered architecture enables the sustained rolling contractions needed to successfully push matter through the extensive plumbing of the human body.
To truly appreciate how vital this system is, we must look at what happens when it fails. A prime example is a condition known as achalasia of the cardia. The underlying pathology of achalasia is the degeneration and death of the vital nerve cells within the Myenteric or Auerbachs plexus at the lower end of the esophagus. Because the smooth muscle is no longer receiving the proper neurological signals, the mechanics of swallowing break down. Specifically, it results in the absolute failure of the cardioesophageal sphincter to relax when food approaches. The result is a severe motor disorder. Patients experience a complete loss of peristalsis in the esophageal body. Because the sphincter stays tightly clamped shut and the esophagus cannot push downward, food is prevented from entering the stomach, causing it to back up. This clinical correlate highlights that smooth muscle health is entirely dependent on the delicate nervous plexus that governs it.
Our final clinical correlate highlights a rare genetic disorder known as multisystemic smooth muscle dysfunction syndrome. This condition vividly illustrates just how ubiquitous smooth muscle is across the body. The genetics behind the syndrome involve a rare mutation in the ACTA2 gene. This specific gene is responsible for coding smooth muscle alpha-2 actin, which is the vital contractile protein necessary for movement. When this protein is malformed, the smooth muscle fails system-wide. Visually, we see ocular issues where the pupils become fixed and dilated, exhibiting a decreased response to light due to ciliary muscle failure. Internally, there are severe vascular abnormalities because the smooth muscle lining the blood vessel walls cannot properly maintain tone. Finally, patients suffer from gastrointestinal and renal complications due to a dangerous weakening of the bladder and the entire GI tract. This syndrome is a powerful reminder that while smooth muscle works silently, its health is absolutely fundamental to human survival.